All right, so let's go over paper 6, which is the alternative to the practical for biology. That's IGCSE from Cambridge International Examination, CIE, October-November 2016, so that's winter 2016, variant 1, so 0610/61. All right, let's get started.
Right, let's get it done.
Question 1. Some animals have a body temperature that is higher than the temperature of the environment. As a result, these animals lose heat to the environment, causing their body temperature to fall. An investigation was carried out to find the effect of the volume of the body on the loss of heat to the environment. The volume of the body of an animal and its temperature can be represented by hot water. So there's 5 steps in this experiment, a lot shorter than some of the experiments. So step 1, you take 250 centimeter cube beakers were labeled A and B. Step 2, a line was drawn on beaker A, 6 centimeters up from the bottom of the beaker, and a line was drawn from beaker B, 3 centimeters up from the bottom of the beaker. So we have 2 beakers. 1, 2, beaker A and beaker B. And on beaker A, we have a line drawn at 6 centimeters, and beaker B, we have a line drawn at 3 centimeters. Okay, hot water was added to both beakers up to these marks. Step 4, a thermometer was placed in the water in each beaker and a timer was started. The temperature of the water was measured immediately in both beakers and recorded in a results table. The thermometers were left in the water throughout the investigation. Step 5, the temperature of the water in both beakers was measured and recorded every minute for 5 minutes. Okay, the results of this are on the next page. A. Prepare a table in the space provided to record these results use figure 1.1, which is on the next page, to complete this table. So whenever you record a table, what you're changing, the independent variable, is always in the first column, and what you're measuring is in the columns after that. All right, so the independent variable here is whether it's beaker A or beaker B. So we have beaker, and we have the next column is time, and it's being measured once a minute for 5 minutes. So it actually means it's best to have units in minutes. Normally, you want the units in seconds. That's when you're measuring it. This is just saying when you're measuring it, not it is the measurement. The time is not the dependent variable. Time is just when you're measuring it, and it's once a minute. So you can keep it in minutes. And the third column is temperature. And temperature is in degrees Celsius. Okay, remember you have to put the units in the headings. Don't put any units in the table. All right, I always like to write in what's in the first column first, and then I draw on all the cells around it. Then I know that everything next to it lines up. You might have a different method of doing it. There's no real right way of doing it as long as it doesn't look messy. Okay, so I have 6 readings for A and 6 readings for B. The reason why I have 6 is because it's measured once every 5 minutes, but we have a starting time as well. Okay, so now we have all the cells drawn in, and we know that they'll line up nicely. All right, so now we put in the time. So we know each one was measured once a minute for 5 minutes with a starting time of 0. So 0, 1, 2, 3, 4, 5, 0, 1, 2, 3, 4, 5. Okay, so that's the time in minutes that it's measured. Okay, now we have to look at the temperature. Okay, so here are the different thermometer readings. We have 10 in total. We have beaker A at the top and beaker B at the bottom. And each mark on the thermometer is 1 degree Celsius. And if we look at this, it looks like pretty much all of these lines for the thermometer go up pretty much exactly to the mark, except this one at 5, where it goes halfway between them. So this is 70.5 degrees Celsius, which means we should probably do them all to 1 decimal place. Okay, so this one is 83 degrees Celsius, 83.0 degrees Celsius. This one is 81.0 degrees Celsius. This one is 78 degrees Celsius. Okay, this one is 76 degrees Celsius. And this one is 74 degrees Celsius. 74.0 degrees Celsius. Okay. So if you look at the numbers for beaker B, this is 83.0 degrees. 83.0 degrees Celsius. This is 76.0 degrees Celsius. This one is 72 degrees Celsius. And this one is, this is 68.5. Again, it goes halfway. 68.5 degrees Celsius. And this one is 64 degrees Celsius. And this one is 63. All right, let's write those in the table. All right, these are the numbers recorded in the table. Make sure your decimal points are nice and clear, and you probably wouldn't lose marks if you didn't have them all to the same decimal place, but it's good practice to put everything to the same number of decimals. In A level, they'll be very picky about that. B1, the rate of heat loss is the fall in temperature per minute. Calculate the rate of heat loss between 0 and 5 minutes for both beakers, and include the units. Show your working. So beaker A, the rate of heat loss equals the temperature difference, the starting temperature minus the ending temperature, divided by the time. All right, so it started at 83.0 degrees Celsius, and it ended at 70.5 degrees Celsius. And you divide it by 5 minutes. So the answer, if you calculate that, is 2.5 degrees Celsius per minute. So you write in here 2.5 degrees Celsius per minute. If you don't write in the units, you don't get the marks, because also here it says very clearly in bold, include the units. Okay, so the rate of heat loss in B. So rate of heat loss equals, it is 83.0 degrees Celsius minus 63 degrees Celsius. So the rate of heat loss here, divided by 5, is 4 degrees Celsius per minute. So the answer is 4 degrees Celsius per minute. All right, that's it. 2. Using your results, suggest a relationship between the volume of the body and heat loss. Okay, so here, using your results. What that means is you'll probably get a mark for quoting data. Okay, so what we can look at here, what we can see with our result, is that beaker A, that had a larger volume of water in, to represent the larger volume of the body, lost a lot less heat per minute than beaker B, that had a lot less volume in. Okay, so we can say that the greater the volume of the body, the smaller the rate of heat loss, or you can say it the other way around. So the greater the volume of the body, the smaller the rate of the heat loss, or the other way around. Now we have 2 points for this, and it says using your results, so just quote some data. So you only have 2 bits of data you can use. So you can say the rate of heat loss in beaker A was 2.5 degrees Celsius and the rate of heat loss in beaker B was 4.0 degrees Celsius. Remember, put in the units and you should be fine. C, state 2 variables in this investigation that have been controlled. All right, so the one of the things that's been controlled is the starting temperature of the water. They were both at 83 degrees Celsius. So the starting temperature of the water. Another thing that was controlled was the time intervals to record the temperature. So the time intervals to record temperature. Other things you could have put down, you could have said that the temperature of the environment was controlled because they would both be in the same space. You can say that the volume of the beaker or the size of the beaker or the shape of the beaker is being controlled. And you can say the total time for the investigation. So as you can see, time isn't a valid variable. You can say time intervals to record the temperature or the total time because those are different things. All right, but you only put down 2 because the question only asks for 2. It even says it in bold letters. Good. 2, suggest why the thermometer must be left in the water throughout the investigation. Okay, the reason why it must be kept in the water is because it actually takes time for the thermometer to adjust to a new temperature. And if you put it in every minute, you won't be reading the temperature at that minute. You'll be reading the temperature as it's adjusting. So it takes time for the thermometer to reach the water temperature. 3. There is a possible source of error in step 2 of the investigation. Identify this source of error and describe how to modify step 2 to improve the investigation. All right, let's look at step 2. Okay, so step 2 was when they drew a line on the beaker. That is step 2. Okay, so the thing that is a possible source of error in this was that they measured the height of the line. They didn't measure the volume of the water. Another thing you could say is that it's difficult to draw the line accurately, or it's also difficult to judge the water level against the line. Those are all valid things you can say. And to improve this investigation, the best way to cancel out that error is to measure out a volume of water using a graduated cylinder. So a possible source of error was that the height was measured and not the volume. To improve this investigation, you can measure out the water volume using a graduated cylinder. All right, and now suggest one safety precaution students should take while carrying out this investigation. Well, there's lots of things you can do. You're using hot water, so you can say something like, wear goggles to protect against splashes. So wear goggles to protect against hot water splashes. You can also say something like wearing gloves, using tongs, wear a lab coat, and standing up throughout the investigation, because if you're sitting down, it takes longer to move out of the way if something's about to spill. All right. D. Some students were asked to test the hypothesis. The colder the surroundings, the faster a small mammal's temperature will drop. Describe how the student could modify the investigation described in steps 1 to 5 to test this hypothesis. Okay, so instead of testing the volume of water, the body size, they're saying how warm or cold the surroundings are. Okay, so remember, in an investigation, you need to have an independent variable. You need to have a dependent variable. You need to have controlled variables. You need to have something about the method. You also really should put in something about reliability. That is, repeat and take the average, or repeat twice and take the average. Okay, and you can sometimes put in something about the results. What are you actually looking for? Okay, these don't have to be put down in that order, but when you're done, when you've run out of ideas, just go back and check to make sure you have everything that you're supposed to. Okay, so the first thing you want to do is you want to take 2 or 3 identical containers. Just choose a number and you can use that. If you said 2, that'd be fine. If you said 5, that would also be okay. All right, so another thing you do is add some water and make sure it's at the same temperature. So add 250 centimeters cubed of water at 85 degrees Celsius to each container. Just choose some numbers that sound reasonable. And another thing you can do is, after that, you want to place those containers into 3 different temperatures. So place containers in areas of 3 different temperatures, one in a fridge, one at room temperature, and one in a hot water bath. Then you want to measure the temperatures of each container at 1-minute intervals. So you want to measure the temperatures of each container at 1-minute intervals. You can also time how long it takes for the temperatures to reach a certain level, a certain number of degrees that you state what it is. So you want to repeat twice and take the average. That's your reliability. And then you want to compare the rate of heat loss at each temperature. So compare the rate of heat loss at each temperature. So let's double check. Do we have an independent variable? Do we have something that we're changing? Okay, the independent variable here is you are changing the temperatures that the containers are in. Your dependent variable, the thing you're measuring, is you're measuring the temperatures of each container at 1-minute intervals. Controlled variables, well, we have 3 identical containers. We have 250 centimeters cubed of water, 85 degrees Celsius. That's 3 controlled variables there. Do we have a method? Well, we are describing the method in this process, so yes, that counts. Reliability. The reliability is repeating twice and taking the average, and we have something about the results. Okay, so this is, you don't have to have something from everything. Sometimes you could put in something about safety. Sometimes you could put in something about what is the control experiment where you expect nothing to happen. But you want to get things from as many different sections of an investigation as possible. Definitely independent, dependent, controlled, method, and reliability are the key basic ones. Okay, after that, there's a few other ones you can sometimes include. And that is a 6-mark question. 1, 2, 3, 4, 5, 6. But add 250 centimeters cubed of water, 85 degrees Celsius, that's actually 2 marks. So you have plenty there. E. Humans sweat when they get too hot. The effect of the temperature of the room on the average rate of sweating was investigated. The results are shown in Table 1.1. Okay, this is Table 1.1. If the temperature gets warmer, the rate of sweating seems to get higher. 1. Plot the graph using the data in Table 1.1 on the grid. Okay, so this is the data you need to plot. Now, if ever you are unsure of which one to put on the x-axis and which one to put on the y-axis, well, whatever is in the first column, that should be the independent variable, as we said before. And the independent variable should always be on the x-axis. So the y-axis is the dependent. So that should go on the y-axis. Okay, that's just to make sure you're clear. Now, for temperature, you need to go from probably 0, you need to go between 13 and 40. Okay, so probably 0 to 40. And for the rate of sweating, you probably need to go from 0 to, let's say, 1200. That's a reasonable number. So let's look at our axes. Okay, first of all, we're going to label the axes. So temperature of the room in degrees Celsius on the x-axis, and the average rate of sweating in centimeters cubed per hour on the y-axis. Sadly, the whole graph doesn't fit on the screen, but we'll work with what we have. Okay, so they've given us 7 large squares. This is a large square. That's one large square. And we need to go up to 1200. So a reasonable number to go up by is 200. So we go 0, 200, 400, 600, 800, 1000, 1200, and we can go up to 1400. But so we're going to be using just above 1000, and that's well up over half the graph. Now the y-axis is a bit more confusing. I don't like the number of boxes they've given you, the large squares. They've given you 6 large boxes. So 1, 2, 3, 4, 5, 6. And you need to go up to 40. So if you can do it 2 methods. Okay, you can say 0, 10, 20, 30, 40, and then you could always write 50 and 60 down here, but you will only use this amount. It's not using the whole graph, but it is using more than half, and I think this is the one that they expect you to use. Okay, I don't like it as much because there's a large part. If they'd just given you 5, then it would have been much more clear. Okay, so that's one way you can do this. I've had many people ask, do you actually have to start from 0? And you don't actually have to start from 0. You can put this little squiggly line here and make this 15, 20, 25, 30, 35, 40. And that uses up much more of the graph. It looks much better, I think. And 13 would be right here. The starting number would be right here. Okay, I'm going to show you the first way, which is the way I think that they meant, because there is a little trick to it. Okay, so we have 0, we're going up by 10s. It's nice and easy to graph, but there is a little trick at the end to this. Okay, so let's plot the points. And just to be clear, you're going up by 200 in 10 squares. So 200 divided by 10 equals 20 centimeters cubed per hour per square. Okay, you don't need to show this. This is just so you know how to work out how much each square is. Okay, so these are the points. If you notice, I drew them with little X's. The examiners like to see X's or little dots, small dots with a circle around it. They will mark you down if you do a big blob. They'll mark you down if you do a little tiny dot. Okay, because the little tiny dot will disappear. when you go over it. And the big blob, well, it's not very clear exactly where you're talking about. All right. Now you have to draw your line of best fit. Now this one, obviously, you can't really use a straight line with a ruler because it's very curved. You could do join each point with a ruler dot to dot. That'd be okay. It's also absolutely fine, probably better to very carefully freehand. You can see that these actually all line up. This is why you do it in pencil, because it's very difficult. I'd erase this bit here to join them together. You do want all the points joined together. Make sure you use a sharp pencil, okay, when you do this. That's very important, because if it's too blunt, if it's too dull, then the line is too thick, and they might actually miss some of these points. All right. Okay, that's a terrible line, but it shows the trend of the graph. Okay, as I said before, there is one trick to this graph, which you can't see if you take up the entire graph. The point ends here. You cannot continue the line straight up. You can't just assume that that's the way it's going to go. If you draw this section here, you will lose a mark. Because, in theory, the line could start going flat. Okay? You don't know what it's going to do. So you can't draw a line there. Your line of best fit should only include the points that you know. 2. Describe the effect of the temperature of the room on the average rate of sweating. Okay, now it doesn't say use your graph to describe the effect of the temperature of the room on the average rate of sweating, because if it said use your graph, then you would probably get a point from just quoting a data point. This is just describing the effect. Okay, so the effect is that as the temperature goes up, the rate of sweating goes up. So as temperature increases, the rate of sweating increases, but that's only one mark. Okay, the other thing we can see is that it's not increasing in a linear manner. It's actually increasing exponentially. So the rate of increase is increasing. So the rate of increase is not a linear relationship. You could say the rate of increase is increasing, or you could say the rate of increase is exponential. They're the same thing in this case. Okay, next. Question 2. Figure 2.1 shows photographs of 5 different flowers, A, B, C, D, and E. Some of the structures of the flowers are labeled on the photographs. So we have petals, stigmas, anthers, and filaments. They don't all come up at the same time. That's okay. The genus of the 5 flowers can be identified using this key. Okay, so we can look at each flower, use the key, and figure out what it is. So let's start with flower A. Are the petals present and large? I would say yes, they are definitely very present and large. So we go to 2. Do they have 4 petals, or do they have more than 4 petals? Okay, I see 1, 2, 3, 4, and more. So there's more than 4 petals. So they have more than 4 petals. We go to 3. Is the stigma clearly divided into 5? And the stigma here, 1, 2, 3, 4, 5. Yes, it is. So that means if the stigma is clearly divided into 5, this is a geranium. So we'll fill in the letter, this question A, use the key to identify the flowering plants, A, B, C, D, and E. Letter A is a geranium. All right, now we'll go to B. So again, are the petals present and large? Well, no. No, they're not. Certainly if they're present, they're not very large. So the petals are absent or very small. So we go to 4. So are the anthers large and hanging on long filaments away from the flower? These are the anthers, and yes, they're hanging down and they're quite large. So that means that this one is sorghum. Okay, so B is sorghum. All right. Now we go to C. Again, are the petals present and large? Well, those look like very nice large petals. Yes. So the petals are present and large. We go to 2. Do they have 4 petals or more than 4 petals? 1, 2, 3, 4. I see 4 very nice petals. So 4 petals mean this is draba. Okay, this is draba. All right, D. Okay, again, are the petals present and large? they are, yes, there's petals. You can definitely see them, and some of them are larger than others, but yes, they look pretty big. Okay, so presents, they have petals that are present and large. Go to 2. Do they have 4 petals or more than 4 petals? Certainly I count more than 4 petals here. I don't know how many there are, but there's certainly more than 4. Okay, more than 4 petals, we go to 3. Is the stigma clearly divided into 5? Or it's not clearly divided? Well, it's difficult to see the stigma, but it's certainly not clearly divided from this picture. So I'd say it's not clearly divided. So that means if it's not clearly divided, that it's a fuchsia. Okay, so D is fuchsia. And then E. Again, are the petals present and large? Nope. If the petals are there, I don't know if those are petals or not, but they're very tiny. So the petals are absent or very small. Go to 4. Okay, so the anthers large and hanging on long filaments away from the flower? No, they are not. So let's see what it is. So that means E is dactylous. Dactylous. Okay, that is the answer to this question. B. Figure 2.2 shows 5 stamens, the anthers and the filaments, from an insect-pollinated flower, Gloriosa superba. Oh, that's a great name. Gloriously superb. I like that name. Gloriosa superba. Let's see what the question asks. And here's the part where it says, 1. Make a large labeled drawing of the stamen enclosed by the box on figure 2.2. There has to be a drawing somewhere on every biology alternative to the practical exam or practical exam. So key points when you're doing a drawing in biology, use a sharp pencil. Sharp HB pencil really is best because HB pencils are easy to erase. But a sharp HB pencil, make sure you have a good eraser. And you want to make sure you have smooth, clear lines, uninterrupted. So you want to make sure that's nice and smooth. One smooth, clear line. You should make it so that you can't really tell the difference where you start and end. So if you go like this and go like, that's actually pretty better than I normally do. If you draw it quickly and go like that, you will lose a mark. Also, you can't have any hatching. You can't do any shading. You can't do any stippling to show texture or anything like that. You can't do anything like that. Basically, you're just drawing the outline. That's all they really want for these. They do not want any more detail. But you need to draw in the same proportions and direction as the original, as best you can. The examiner knows you are not an art student. Well, you might be an art student, but in this exam, you are a biology student. The examiner knows that. So as long as you've done a good job of trying to put things in the same proportion and same directions, you'll probably be okay. The other thing is never, ever, ever use a ruler on a drawing unless really there's definitely a need for a ruler. But in biology, you don't use a ruler for drawing. You do use a ruler when you label though. This is one of the few ones where it says draw a labeled diagram. What do you label? Okay, so this is the stamen. The stamen is made up of 2 parts. It even says up above here, it says 5 stamens, the anthers and the filaments. So you need to label the anther and the filament. Now, do not ever draw, when you do a label line, do not ever draw an arrowhead. That is wrong. You need to use a ruler for this. You need to draw a straight line with a ruler, and you have to make sure that the lines don't cross. Okay, so that's a reasonable drawing. It's not great, but it will probably get you the marks you need, other than I'm missing the labels because it says a large label drawing. So let's label this drawing. Okay, so we have 2 things we're labeling. We're labeling the anther and the filament. So I'll do a line to the filament, and I'll do a line to the anther. Okay, now we just have to label those. So we have the filament and the anther. The lines do not cross. You just have the line touching the structure that it's supposed to. There's no arrowheads. So what do you get marks for on this drawing question? 1, you get a mark that is just the outline. You haven't done any shading, any stippling, any hatching, or any well, you can't really tell the difference between start and ends of lines. And that's important. That's why you don't want to draw really hard with your pencil. You want to be able to erase it. Okay. Another mark is if it's the right size. You don't want it too small, so it has to take up at least half the space. Too big would mean it would be actually over some of the writing. You don't want it over the writing. The detail, you want to make sure that you drew it to approximately the same proportions as the original. So you have your anther, you have your filament in the same, facing the same direction, approximately as in the original, and the anther is about half the size, half the length of the filament. You can use your ruler and double check that it's approximately half the length. And approximately half the length, not exactly half the length. And it asks for a label drawing, so you need your anther and filament labeled. Okay. 2. Measure the length of the filament on figure 2.2 and include the unit. The reason why it says include the unit is it's better to do it in millimeters, but it's okay if you do it in centimeters. That's absolutely fine. So it wants you to take your ruler and measure the length of the filament. You want to make sure when you do your measurement that you've drawn on the line that you're measuring. Do it in pencil. I'm just doing it in red so you can see it better. Okay, draw the line and draw the start and end, and then measure along that point. Then they know that what you measured. Okay, because I printed it out on a slightly different size paper, I measured 27 millimeters. But anything on this one, anywhere between 27 and 31 millimeters is okay. But if you're printing it out on a different size piece of paper, you might have a different measurement. That's okay. Just you can either work with what you have or what's in the mark scheme. But as long as you know how to measure it, that's okay. Okay, so 27 millimeters is what I measured. So the length of the filament on figure 2.2 is 27 millimeters. Okay, you could do it in centimeters as well, but that's why they say you have to include the unit. Measure the length of the filament on your drawing and draw a line on your drawing to show where you measured the filament. So the same line that we used on the other one. You don't have to necessarily do it on the original diagram, but it's better to. Again, I'm just using red to make it clearer for you. So we're measuring along this line here, and this is the end. And this is the end, just in red to make it clearer for you, but you will draw it in pencil because that's what you need to do. And when I measured this, I measured 65 millimeters. So I measured 65 millimeters. Okay, so now the magnification, the formula for that is the length of the drawing divided by the length of the actual measurement. So calculate the magnification of your drawing using the information above and your answers. So magnification equals drawing divided by actual. Okay, so that is 65 millimeters divided by 27 millimeters, and that equals 2.4 millimeters. Okay, 1 decimal place is all you need. Okay, maximum. So 2.4 millimeters is a good number. If you do not ever, for magnification, put in more than that, because it's a drawing, it's not as specific. You might have, if you had measured the anther, it might be a slightly different number. You're doing it as best you can. So never do it too precise on magnification. So you can say it's times 2.4. And now we've reached the end of this exam. I really hope you found this helpful, and if you have, please press the like button, the thumbs up, and we'd appreciate it if you'd subscribe if you haven't already. We love to hear from you, so if you have anything to say to us, then please write it in the discussion section below, and have a great day.